Hi, my name is Rebecca Pinals, and I work with Professor Marquita Landry at UC Berkeley. Today, I will be sharing our very recent work in how we can harness the protein corona towards carbon nanotube-based sensor design, and specifically for developing a SARS-CoV-2 nanosensor. Diagnostic testing to identify those infected with SARS-CoV-2 is crucial to control the spread of disease. Here, I am showing a map that displays CoV-2 tests conducted per new confirmed case. The WHO has suggested around 10 to 30 tests per confirmed case as a general benchmark of adequate testing. As we can readily see, however, many countries are not meeting this target in testing. And so while testing is key to controlling this pandemic, diagnostics still suffer from insufficient supply, throughput, as well as accuracy. Briefly, we can categorize current COVID-2 tests into three main types. Molecular tests, such as PCR-based assays, remain the status quo for active infection diagnosis, yet require trained personnel, specialized equipment and reagents, clinical sample purification, and longer turnaround times. Antibody or serological testing provides important surveillance data, but not infection at the time of the test. Antigen testing has now been emerging as a rapid diagnostic alternative, looking for active virion components in patient samples. However, these are currently less sensitive tests. In light of the current COVID-2 diagnostic limitations and ongoing pandemic, we asked ourselves, can we develop a platform orthogonal to these current types of testing to detect active COVID-2 infections that is rapid, accessible, and reliable? In the Landry Lab, our expertise is in developing carbon nanotube-based sensors for biological analytes. We employ single-wall carbon nanotubes towards this end due to their numerous advantageous physical and optical properties. These carbon nanotubes are at the nanoscale and possess a very high aspect ratio. There are various methods by which we can modify this large surface area to render these carbon nanotubes selective to specific analyte binding to build a nanosensor. And broadly, these are grouped into covalent and non-covalent functionalization strategies. In general, we employ non-covalent functionalization to preserve the underlying graphitic lattice of the carbon nanotube and thus preserve the optical properties that I will introduce in a moment. However, covalent techniques do show different advantages for linking various chemical groups onto the surface. For optical properties, carbon nanotubes possess an intrinsic near-infrared fluorescence in the tissue transparency window, meaning that we can image this signal through various biological media. These carbon nanotubes are also extremely photostable. Previous sensors have displayed reversibility. While our prior work has focused on applications such as dopamine imaging in the mouse brain, as I'm showing here, these same properties lend carbon nanotubes favorably towards viral detection. The carbon nanotube sidewall offers various methods of functionalization to attach sensing moieties, and the near-infrared emission means the signal would be transmissible through non-purified patient samples such as saliva and nasal fluid. I want to now take a step back and say that engineered nanoparticles offer excellent functionality and control in probing and altering biological systems. Yet when we apply these nanoparticles in biomolecule-rich environments, uncontrollable and unpredictable protein adsorption to the nanoparticle surface often prevents effective application of our nanotechnologies. This phenomenon is known as protein corona formation, and I will note that this is different from the coronaviruses we will be sensing, although the name arises from a similar halo or crown-like appearance of proteins on the nanoparticle surface. On the one hand, this protein corona formation could attenuate or eliminate our nanoparticle function by not allowing access of the analyte of interest to the nanosensor surface. However, this corona also presents an opportunity to exploit these either non-selectively adsorbed or selectively attached proteins towards some useful outcome. My prior work has centered around characterizing the interactions between proteins and carbon nanotubes, specifically characterizing the protein corona composition, driving forces, and dynamics. With this project, I propose to harness these innate protein nanoparticle interactions to build a nanosensor. More broadly, this corona-centric design approach is applicable to biological sensing, imaging, and delivery applications. And accordingly, today I will share how we effectively leverage the protein corona to build a carbon nanotube-based sensor to detect SARS-CoV-2. With this goal in mind, We proceeded by taking advantage of carbon nanotubes as our signal transducer, biomolecules with natural recognition abilities for our analyte of interest, the COVID-2 virus, and ultimately we aim to have this nanosensor function in unprocessed patient samples. Returning to the second point, what I mean by this are biomolecules such as the ACE2 receptor, which is the membrane receptor found on our own cells on host cell membranes, to which the COVID-2 virus specifically binds to gain entry. Another example is antibodies used by our immune systems to ward off infection, and we can also take advantage of complementary DNA, which can hybridize with the viral RNA. And towards the last point, as I've already mentioned, the near-infrared fluorescence emission of carbon nanotubes would enable collecting the sensor readout to detect COVID-2 in highly scattering crude biofluids. I am currently leading a research team in developing SARS-CoV-2 nanosensors using several different approaches. We have formulated these designs by taking advantage of the different sensing moieties that possess affinity for the COVID-2 components as I just introduced, and using both covalent and non-covalent attachment strategies to the carbon nanotube surface. In each of these construct cases, we are aiming for some sort of modulation in the carbon nanotube fluorescence upon viral analyte binding. Today, I will just be focusing on the first nanosensor construct that I have been leading the development of, and this is using a non-covalent attachment chemistry. Specifically, the part of the virus that this nanosensor will detect is the spike protein protruding from the virion surface, as shown in dark blue in this schematic. I will be taking advantage of the ACE2 receptor CoV2 spike interactions in formulating this nanosensor design. To construct this CoV2 nanosensor, I employ a passive immobilization strategy, beginning with DNA-solubilized carbon nanotubes. Carbon nanotubes by themselves are extremely hydrophobic and not water-soluble. Therefore, we must employ probe tip sonication with amphiphilic polymers, such as DNA, to suspend these carbon nanotubes in aqueous solution. I chose to then implement this passive corona exchange from DNA to proteins rather than directly sonicating with the proteins in order to maximally retain the protein secondary structure. Further, my prior work has indicated that proteins generally outcompete DNA for the carbon nanotube surface, and we hypothesized that this membrane protein would display innate affinity for the graphitic carbon nanotube surface. Here I'm showing the resulting near-infrared fluorescence spectra, where adsorption of ACE2 onto the carbon nanotube quenches the carbon nanotube fluorescence. This quenching response levels out to about -40%, and this is an extremely time-stable construct with adsorbed ACE2, which is desirable such that our nanosensor baseline is constant. We can then delve in a bit more to try to understand these molecular interactions in forming the nanosensors. I use a corona exchange assay previously developed to monitor the DNA displacement from the carbon nanotube surface by tracking the fluorophore tag on the DNA. Here, fluorophores in close proximity to the carbon nanotube surface are quenched and enter a bright state when desorbed into bulk solution. Now I'm showing that ACE2 adsorption displaces DNA from the carbon nanotube surface, as we hypothesized, in a concentration-dependent manner, as we might expect. Although it may seem that we want to maximize the loading of this sensing protein onto the carbon nanotube surface, I also aim to balance this with other nanosensor properties. To this end, both the absolute values of the concentrations and the ratio between the concentrations of protein and carbon nanotube were optimized, and this yields a mass ratio of 2.5 ACE2 to carbon nanotubes. Interestingly, this ratio was in very close agreement with our footprint calculation as an estimate and was further refined by this experimental optimization. In an effort to further deconvolute the interactions occurring at this protein-carbon nanotube interface, I next employed a surfactant displacement assay. This is based upon addition of surfactant, here sodium cholate, that coats any solvent-exposed carbon nanotube surface and can displace low-affinity molecules from the surface. When sodium cholate adsorbs, this causes exclusion of water from the nanotube surface and produces a large increase in fluorescence and blue shift. Before ACE2 incubation, addition of sodium cholate does indeed elicit a large blue shift and a sizable increase in fluorescence. And here I'm summarizing these spectra as a function of time. Upon passivation with ACE2, these spectral changes were drastically reduced, as I'm showing here. This result suggests a mechanism in which ACE2 adsorbs to the carbon nanotube's surface and stabilizes that surface to further perturbation. Next, we can test how our nanosensor performs in recognizing the SARS-CoV-2 spike protein. And specifically, we are employing the CoV-2 spike protein receptor binding domain, or SRBD, as the viral protein analyte. Upon addition of 10 mg per liter of the spike, our nanosensor undergoes a large increase in the carbon nanotube fluorescence. Shown as a function of time, this reaches a max of 100% increase in signal over the course of 90 minutes. We can further test the nanosensor response to varying concentration of the spike RBD protein analyte, as shown here. I'm now summarizing the response to this spike protein concentration series after 90 minutes in this graph on the bottom right, and we fit to a Hill equation, so a cooperative binding model, to gain estimates for the nanosensor kinetic parameters. Briefly, I've also demonstrated that these nanosensors are colloidally stable, retaining their response to the CoV-2 spike protein after centrifugation, as well as after overnight incubation in ambient conditions. I next measured the nanosensor selectivity to similar viral analytes, including the spike proteins from SARS-CoV-1 and MERS. Here, I'm showing that we do have some nice selectivity to the CoV-2 spike protein. Keeping in mind that SARS-CoV-1 also enters cells through ACE2 spike protein interactions, yet this binding occurs at lower affinity, we see that this is nicely recapitulated with our nanosensor response magnitude. If we next test for CoV-2 spike protein recognition by our nanosensors in varying biofluids, we see that the nanosensor performs best in phosphate buffered saline, mainly retains function in viral transport medium, or VTM, yet displays an attenuated response in other biofluids, most likely due to non-selective biofouling of the complex biomolecules present. To mitigate formation of this unfavorable unintentional protein corona, we employed a passivation strategy with phospholipids that are attached to the hydrophilic polymer polyethylene glycol, or PEPEG, as previously demonstrated in our lab. This passivation enables some retention of nanosensor function in saliva, yet poses a challenge that we are pursuing with ongoing optimization. Finally, I translated this nanosensor platform from an in-solution to surface immobilized format, as we see this as our next step in translating to point-of-care diagnostics. Excitingly, our nanosensors retain their response profile to the spike protein when immobilized on glass slides, as shown by these near-infrared images and traces, where the blue line averages over 12 regions of interest. Also importantly, the sensor response is rapid, maximizing within 5 seconds of analyte addition. And even more excitingly, we were able to test these nanosensor constructs with virus-like particles sourced from our collaborators. And our nanosensors show this robust over 70% increase in fluorescence. As a control, we found that using virus-like particles that do not have the spike protein only resulted in a 20% change in fluorescence, thus confirming the specific spike ACE2 interactions responsible for the sensor response. In summary, we have effectively introduced a new promising methodology for carbon nanotube-based sensor design by harnessing innate protein nanotube interactions. We have implemented this design strategy for the very critical issue of SARS-CoV-2 sensing. And as I have just shared, I've characterized the nanosensor stability, mechanism, various response metrics, as well as how we can adapt these nanosensors into a surface immobilized format. Importantly, the nanosensor readout is rapid and selective to CoV-2. Moving forward, we are pursuing these initial nanosensor designs with ongoing sensor development and optimization. We are excited to expand our testing with more virus-like particles thanks to our collaborators. And next, we will be assessing immobilization strategies into a hydrogel or paper assay format. Ultimately, we envision development of a low-cost point-of-care diagnostic platform with a form factor similar to that of a cell phone for rapid and real-time detection of active SARS-CoV-2 infection. Lastly, I would like to say thank you to my lab, especially my advisor, Professor Marquita Landry, and the whole team of researchers involved on this COVID diagnostics project from within our lab and in collaborating labs. Thank you to our funding, and specifically to the Women in Chemical Engineering Committee for the Travel Award. Thank you for listening, and please feel free to reach out to me with any questions.